The overall goal of this methodology is to evaluate multiple enzymes using a single high-throughput screening system.
This method can help answer key questions in the microorganism and bioengineering field about metagenome screening and enzyme engineering. We have developed genetic enzyme screening system which is consisting of DMPR mutant responding phenolic compound and its downstream GFP reporter. Once any metagenomic gene shows the enzyme activity of our interest, by reacting with the provided substrate, a phenolic compound of the reaction product activates DMPR mutant and trigger GFP reporter expression, which can be easily captured by a flow cytometer. The main advantage of this technique is that, unlike conventional screening techniques, this single method is applicable for screening multiple types of different enzymes by using the appropriate substrate. Demonstrating this procedure will be Kwon, a graduate student in our laboratory.
After constructing a metagenomic library in E. coli with a phosmid vector using a phosmid library production kit, transform the PGES E135K DNA into the metagenomic library cells. Then store the transformed library cells at -70 degrees Celsius in 20 microliter aliquots. To remove the false positives, first thaw a metagenomic library cell aliquot on ice. Then inoculate 10 microliters of the thawed cells in 2 milliliters of lysogenate broth.